All right. So thank you guys for the invitation to speak today. I'm really excited to tell you about some of the work that came out of some of my thesis, as Lucas said, from Stanford in genetics, with a heavy focus on biophysics and how we can apply machine learning methods and where they might fit in best to the overall experimental pipeline of biotechnology. So a little bit more about me. I did my undergraduate degree in biology at the University of Utah, focused pretty heavily on wet lab biology at the time, and then transitioned in 2015 to my PhD in genetics at Stanford, where I made the switch from a primarily wet lab background to a primarily dry lab or computational background, and started learning and applying machine learning methods to the problems that we were facing in our lab at Stanford. And then recently defended my thesis about 3 weeks ago now, and shortly thereafter founded a biotech company called Trident Bioscience to take some of the methods and techniques that I developed at Stanford and transition them to commercial applications for the discovery and refinement of useful proteins. And now I want to start my talk with kind of a very general overview, take you guys back to high school science class and the central dogma of molecular biology. And the central dogma states that the information flows from DNA to RNA to protein, this generally kind of one directional movement through transcription and translation, transcription of DNA into RNA, and then translation of the information contained in that RNA into protein. But that's really only half the story, this bit that you kind of get in high school science class. What happens to the protein is kind of lost in that process of explaining this central dogma. But that protein goes on to actually fold into a three-dimensional structure. And that three-dimensional structure is dynamic with time. It actually changes and the shape shifts so that it can perform different functions for the cell. In addition, you can get these post-translational modifications or chemical group additions to these protein structures that can further modify their function. And then this ensemble of all these different structures are what actually carry out the various functions for a single protein sequence within a given cell. And when I say protein sequence or DNA sequence throughout this talk, what I mean is that each of these molecules are composed of different monomeric blocks. So they're polymers strung together with different Lego-like building blocks in a linear chain. And it's the three-dimensional folding of that linear chain that really determines what the structure and ultimate function is going to be. And in the context of this talk, when I say function, what I really mean is binding. So the binding of proteins to each other, proteins to smaller peptides, so small chains of amino acids, or proteins to DNA or RNA. So you might be asking yourself, why are we really focused on this phenotype or this idea of binding from one protein to another protein or one protein to DNA? And the reason is that this is really how biology controls or tunes the kind of rheostat for different interactions and different cellular processes. And this biological rheostat can be defined by these two main axes. The first being affinity, or how tightly the different interactions take place, and the second being specificity, or how specific a protein recognizes its binding partner. And so if we divide this rheostat into different quadrants, we can start with the bottom left where we have non-specific interactions, essentially proteins and DNA just bumping into each other randomly in the cell. If we then go to the other extreme, a really high affinity, high specificity interaction, we have something like antibody-antigen systems. These systems bind almost irreversibly to each other and with really high specificity. So an antibody can recognize its target protein in a sea of other proteins. If we then move down the affinity axis, we run into transcription factors or proteins that bind DNA to regulate the transcription of downstream genes. So these are the proteins that play an integral role in determining which genes are expressed in which cells. And their binding is really important, but it tends to be pretty low affinity, but relatively high specificity. To fill out the quadrant, we can think about nucleosomes, or these proteins that the DNA in your cells is actually wrapped around. And these proteins play an integral role in actually packing the DNA into the nucleus of each individual cell. And they're a really high affinity interaction, but they have low specificity. They generally bind DNA with lower sequence specificity than the transcription factors. And as you might expect, these types of interactions are really important for a lot of different applications in biotechnology. For instance, one thing that you might be familiar with is CRISPR. CRISPR relies on the base pairing between the RNA as part of the CRISPR complex and the DNA sequence that it's trying to modify. And this has been commercialized by a bunch of different companies. Editas and Caribou are just two of these. Another type of interaction that's particularly important is antibodies. And with all of the COVID-related research, I'm sure you guys are probably more familiar with antibodies than you might like to be. But they're used in a variety of different applications. They're, of course, used as therapeutics, as you've been hearing about lately. But they also are the basis for a lot of different pregnancy tests. So these antibodies are recognizing components in the urine that are indicative of a positive pregnancy. And finally, we have transcription factors, which is where I'm going to start my talk, because these interactions are really important for a lot of applications in synthetic biology. And companies like Amyris or Zymergen, which you may not have even heard of, are really interested in how transcription factors are binding and regulating downstream genes and how they might be able to modify that to regulate gene expression for their desired traits. And so, as you might expect, given the importance of designing these different intermolecular interactions or these kind of binding interactions, we might want a set of tools that would allow us to develop and modify different interactions to suit a particular need. So if we think about what tools we might need to actually design these intermolecular interactions, they kind of break down into four general categories. The first, and the ones that I think you guys came to this talk for, is a model of how these interactions are actually taking place. We then need a method to translate those predicted interactions or the predicted mechanism of the interaction into a way to actually test the hypothesis that's made by that model. And so what this looks like in a lot of wet lab biology techniques is generating a set of sequences, what we call a library, that you can then run through a certain experiment and test the actual outcome of the DNA sequence or protein being produced by that library of input sequences. Taking this a step further, we need to actually test that pool or that library of sequences that we've generated via some experimental method to get a measurement about how well they're performing a certain task. And then finally, and ideally, we would like some sort of method to update our initial model given the new experimental data. And hopefully we're able to build a closed loop system where we can get continuous improvement, a better and better model of how these interactions are taking place and the ways in which we might be able to change them to generate new functionally refined protein or DNA variants. And what I'm going to focus on for the purposes of this talk is kind of this upper corner here. I'm going to define what the model of these types of interactions might look like, and then start to think about how we might generate a method for making these hypotheses actually testable. And the reason that this is such a big problem in biology is that sequence space in general, and especially for proteins, is incredibly massive. It's really hard for us to fathom how large sequence space is and how quickly it grows. So for DNA sequences, there are 4 possible nucleotides at each position. And so as you get a longer and longer chain of these nucleotides, the number of potential sequences that that length of chain can give rise to grows as 4 to the L, where L is the length of chain. So it grows exponentially. For proteins, on the other hand, you have 20 choices per position. And so the number of possible sequences grows as 20 to the L. And so here what I'm showing is, given a certain protein sequence that we want to start with, we can mutate a certain number of positions to all 19 other choices. And here on the y-axis is the number of total mutants that we would be required to screen if we want to do this in a saturation mutagenesis or exhaustive fashion. And unfortunately, our most advanced experimental methods have a limitation of about 10 to the 11th sequences that they can screen in a single experiment. And so this limitation really defines our upper boundary for what we can do in an experimental wet lab technique. So to some extent, we must be selective in the design of these protein sequences that we want to screen in high throughput libraries. And to me, this really defines the best place for a home for deep learning methods, because we have these high throughput screening methods available to us. The cost of any given prediction being incorrect is relatively low. We're not using this to diagnose a disease or to immediately treat a disease, but the value of the informed search through pattern recognition is really high. So if we can recognize patterns in the existing data and use that to inform the sequences that we include in our library, it could be really valuable. And as a case study, I want to present the first project that I worked on during my PhD, which is characterizing and modeling the behavior of transcription factors and how they bind to DNA sequences. So as I mentioned earlier in the talk, these transcription factors bind to DNA, and they do so in a sequence-specific manner. And so when I say sequence-specific manner, I mean that the given protein is binding to DNA with a preference for certain short DNA motifs. An example of one is given here. So this is FO4. It's a transcription factor involved in helping yeast fungi respond to phosphate starvation, hence the name FO4. And the sequence preference is given here. And so the dominant nucleotides are these ones at the middle, CAC, GTG, also known, and you might hear me say EBOX motif. EBOX motif generally refers to this CAC, GTG pattern here at the center. But unfortunately, or fortunately for biology, families of transcription factors often bind very similar sequences. And so here we have a related transcription factor, CBF1, that you can see also likes to bind this CAC GTG motif. And so this raises an interesting question, which is, if they have very similar binding preferences, do they actually just regulate the same genes? Are they fully redundant in their function? And if we look at the gene regulatory networks, so here, the transcription factor is given as these kind of central nodes, and all of the genes that they regulate are the ones connected to those central nodes. And what we see is that there's actually a very small amount of overlap between the genes regulated by FO4 and the genes regulated by CBF1. And so the question that we wanted to answer was, if these core binding motifs, so that CAC-GTG, the EBOX motif, is nearly identical, what differentiates the target binding sites for one transcription factor over the other in the actual genome of a living organism? And so what we have to understand is that these binding site sequences sit in different contexts within the genome. They're not just floating out as a 6 or 8 mer sequence in the cell. They're actually connected to a bunch of other DNA, and that DNA has its own sequence. And so our hypothesis was that it might be these sequences that sit outside of that main motif that actually govern whether the transcription factor is going to bind or not. And so we wanted to look at all possible arrangements, so all possible genomic configurations for these given EBOX motifs, and then measure the binding for FO4 and CBF1 against that target library. And the way we did this was we made this DNA library that we're able to use high throughput sequencing on. So we're able to actually measure and read out each of these individual sequences, where we fixed that EBOX motif at the center, CAC-GTG here, and then we allowed the bases next to it to be any of the 4 other nucleotides, so ACG or T. This generated about 1 million sequences in total, and represents an attempt to characterize binding that's actually relatively large. So we don't have a lot of really good methods to measure 1 million sequences with the resolution that we wanted to. And so along with a postdoc and another grad student in the lab, I developed this BET-seq method, which stands for binding energy topographies by sequencing, and it allows us to measure these transcription factor binding specificities in high throughput, importantly with energetic information behind it. So it has to be a biophysical measurement in order for us to be really interested in Polly's lab, my PI's lab. And so we wanted to do this with really high resolution and generate these physically meaningful values for each measured transcription factor DNA sequence interaction. And the way that we did this was we have a small microfluidic device that you can see here on the left-hand side. And this device is about the size of a postage stamp and has a bunch of these inputs and these hydraulically activated valves. So here's kind of a schematic of how the individual chambers within this microfluidic device look. And each one has what we call this button valve at the top that allows us to compress and actually stop the interaction of a transcription factor with a bunch of different DNA sequences. So what this allows us to do is bind transcription factor to the surface here, flow in DNA. We're going to select for the things that bind. They're going to bind. We're then going to trap them and separate them from the sequences that don't bind. And so this separation is what allows us to make a measurement, an energetic measurement of the affinity of different transcription factor DNA interactions. So then we can put each library, so here we have the bound and the unbound libraries, go through high throughput DNA sequencing on a machine made by usually Illumina. And then this generates a library where we can actually order these sequences based on the preference of each given transcription factor to these kind of context sequences outside the eBox domain. And so here you can see we can potentially look at all of these DNA sequences in the library and rank them from highest affinity, which in delta delta G space, so delta delta G is just a measurement for how high affinity a sequence binds, and it has a physical meaning behind it. And the more negative that value is, the more tightly it's bound to the transcription factor. So more negative equals tighter binding, more positive equals looser binding. And so we can do this for the entire library of sequences. But unfortunately, as I said before, the methods that already exist for doing this don't really scale well to the million sequences that we have in our library. And the reason is because experimental noise really inhibits these energetic measurements. The two reasons behind that are that we can't calculate a binding energy for something we don't observe. So for a million sequences, you actually have to use quite a bit of sequencing in order to see every sequence in that library. And for low count values, so things where we only observe a few of those, of that individual sequence, a few of those molecules, the values are extremely noisy, which make our ultimate energetic measurements really noisy. And so we thought about this for a while, and the solution that we came up with was to simulate this process. So the advantage of working in a biophysically meaningful space is that we know how these things should interact in a perfect world, and we can just simulate the experiment and see how well we do for different depths of sequencing. And the important thing to recognize here is that as you go to a deeper depth of sequencing, you're getting more reads, you're getting better data, but it's also more and more expensive. And so we simulated one of these BET-seq experiments, and here on the bottom, we get the mean reads per sequence, so the number of times we're observing each individual DNA sequence. And then on the y-axis here, we're showing the median value for two different measurements of success. And the first is the fraction of the library that we're observing. We want to be able to see all million sequences. And then the second is the R-squared value with the true defined value that we put into the simulation. And we can see that as we start out on the left, we end up with these really kind of garbage data sets where we're only observing a fraction of our library, and the measurements that we're getting don't actually correlate that well with reality. On the other side, we have a really expensive experiment that yields beautiful data where we're observing nearly all of the library. And our data quality is really good as well. The R-squared value is really high with the expected values. But we figured that we might be able to sit kind of in this Goldilocks zone here in the middle where we're observing almost all of our library or all of our library, but the R-squared values might not be perfect. And so that really set up the expectation that if we sequence one of these libraries, if we perform the experiment at really low read depth, what we're going to get out is kind of not reproducible and is going to look kind of like a circular pattern. And in fact, when we ran the experiment, we ran it twice, two different replicates, and we do see this indicative circle pattern where each measurement does not correlate with the same measurement made in a different replicate. But since we had the simulation data in hand, we were able to say that we expected that, made the change to a higher read depth, and we end up getting better data out. But again, the problem with this is that if we run all of these experiments at really high read depth, it's going to cost us a lot of money. And we'd really like to avoid that. So we thought to ourselves, we might be able to use some type of modeling to extract and separate the signal in the data from the low read depth experiments from the noise. And so we wanted to see if we could do that. And the thought behind this was that we already use transcription factor binding models. In fact, I presented one earlier in this talk. So this motif that I showed you at the very beginning, it's important to ask how we actually got to that point. And it's through a very simple linear model that we can go from a bunch of different sequences to one of these motifs. So we asked, how do we get here? Someone previously had run a bunch of DNA sequences, did the same kind of enrichment step that we did earlier, pulled out a bunch of sequences that bound to the transcription factor, performed alignment, and then noticed that in all of the sequences that bound, there was a common pattern, this CAC-GTG pattern, and potentially this flanking nucleotide here upstream and downstream. And from that, they were able to generate what we call a consensus binding site. So a single sequence that is generally enriched in the binding sites for these transcription factors. But an important assumption in this methodology is that the contribution of each nucleotide to binding is totally independent. And so we didn't think that that assumption was that good. We wanted to ask, is this assumption of independence actually valid? And we knew, because the time that we were doing this work, neural network models were coming into vogue, we knew that nonlinear models would enable us to do the same type of modeling, but in an assumption-free fashion. So we wouldn't be forced into assuming that the contribution of each nucleotide in the binding site was contributing independently. And so we built ourselves a nice little neural network model. We feed in the DNA sequence here as a one-hot encoded vector, have a couple intermediate layers with nonlinearities, and then we're going to output the predicted delta delta G value and train on our experimentally measured values. And when you work on deep learning in biology, you run into a bunch of people that aren't too keen to trust that your magic neural network box model is actually accurate. And so you have to do something to prove to them that the data that you get out actually reflects reality. And we had another grad student in the lab, Arjun, who was kind enough to measure a bunch of the sequences that we had looked at on an orthogonal platform. So based on the same technology, but a much higher resolution technique that's much lower throughput. And what he was able to find and show was that if we take our unprocessed or raw data, the measurements that we had made without any type of correction, they don't correlate super well with the measurements that he had made in this orthogonal method. But if we first run it through our neural network and take the prediction output from that and compare it to his orthogonal measurement, they do correlate really well. So this gave us a lot of confidence that what we were seeing was good separation of signal from noise. But we also understood that in exchange for the flexibility of neural network models, we pay a price in interpretability. So we're not able to have that nice little motif anymore where we can say, okay, these are the contributions to binding. And so we were looking for a way around that, and we wanted to take a look at how well each of the existing models that were based on principled biophysical approaches can explain what's learned by the neural network models. So we basically wanted to ask, how far can a mononucleotide model, or that kind of binding motif model that I showed you in the very beginning, how far can that get us in explaining what the neural network has learned? And so if we plot the mononucleotides, the linear model prediction down here on the x-axis and the neural network prediction on the y-axis, we see that for FO4, that linear approximation is actually pretty good. It explains binding behavior pretty well. But for CBF1, on the other hand, it doesn't do so well. There's a lot of variability for any given mononucleotide predicted delta delta G that can't be explained only by those mononucleotide effects. And so there's another method for doing this that adds a little bit more biophysical information, injects a bit more domain knowledge into this modeling process. And that's considering dinucleotide effects. And dinucleotide effects are just combinations of different mononucleotides at different positions. And these dinucleotide effects are really important for a bunch of different functions. But there's reason to believe that for FO4 and CBF1 especially, they might be important. And so what I'm showing here is the actual structure of the FO4 protein, which are these kind of blue squiggly lines up here, and then the DNA that it's bound to, the green helix down here. And I'm showing kind of two views of that offset by 90 degrees. And there's two different types of dinucleotide effects that we wanted to consider. The first were adjacent or nearest neighbor dinucleotide effects. These effects are a single base and the base pair right next to it. And these are really important for determining the actual three-dimensional shape of DNA. And so I know that I've been presenting DNA here as a linear sequence of these letters, but it actually exists in your cells as this three-dimensional helix that can bend, rotate, compact, expand, and a lot of those shape properties are determined by the nucleotides and how they stack right next to each other. On the other hand, we realize that these gapped nucleotides, or nucleotides that are present, dinucleotide effects that are not present in adjacent positions, might be really important because FO4 and CBF1 are actually dimer proteins. And so there's two individual protein chains that bind together and co-bind that DNA complex. And because these tails of FO4 and CBF1 sit within these grooves of the DNA, the contacts that are distal to one another might be actually important in the binding process. So if we add these dinucleotide features to our linear models and then compare them back to the neural network models, What we see is that for Fo4, it nearly perfectly explains binding at this point. There's very little variation left. And for CBF1, it does a pretty significant job, indicating a significant improvement on the explanation of binding. If we add in all of the dinucleotide effects, at this point, we can explain nearly all of the variability in the predictions from the neural network for Fo4, and the same is almost true for CBF1 as well. So what we found from this work was that Fo4 and CBF1 clearly bind in these two very different modes. So one has a strong preference for the mononucleotide effects, so it seems to be that each individual nucleotide is actually contributing independently to binding. But CBF1, that doesn't seem to be the case, indicating that for CBF1, there might be a much larger effect of something like DNA shape to how it binds. But if we want to ask the question why, the work that I've presented so far doesn't really get us there. And we're a bit limited going from DNA sequence to functional predictions directly via a neural network model. And the reason for that, if we go back to the initial presentation of the central dogma of molecular biology, is that instead of predicting each step along this process from protein to the folding and conformational dynamics, the post-translational modifications, and then binding, we're basically black boxing this entire intermediate step and going directly from the protein and DNA sequences directly to their functions. And it's important to note here that we're not explicitly modeling the structure of the protein or the DNA here. We're just taking a proxy measurement of their sequences for their structures. But why would we want to model the structure at all? Aside from the fact that I just told you that it was critically important, and I chose an example from the literature that I think illustrates this really well. And the principle here is that structural changes in the protein actually drive really important functional effects for the protein being targeted. And so here what I'm showing on the left-hand side is green fluorescent protein, also known as GFP. And this is a really important protein in molecular biology because if you hit it with the right wavelength of light, it glows green and it allows us to visualize proteins and motion within the cell. And on the right-hand side are two different nanobodies. So these are really cool single domain antibodies isolated from camelid species. So llamas, alpacas, camels, they have these really cool single domain antibodies. And a lab in Europe isolated two of these different nanobodies that bound this GFP protein. And they were doing some basic characterization of this, and they realized that as they added more and more of each nanobody, they actually saw a very real functional effect. And so the one on top in red, as they added more nanobody, it suppressed the fluorescence of GFP. But the one on the bottom, as they added more and more of it, it enhanced the fluorescence of GFP. And so they named these the GFP minimizer and enhancer nanobodies respectively. And all they really differ in is the site on GFP that they're binding. And what's interesting about this is that if we look at the GFP minimizer and enhancer nanobodies and start to ask, like, okay, what are the functional effects? What are the structural reasons behind these functional effects? We can look at the contact map of GFP. So if we look at all of the amino acids by all of the amino acids and then just highlight the ones that are in contact with each other, we see that when the GFP minimizer nanobody is bound, this is what the contact map looks like. When the enhancer nanobody is bound, this is what the contact map looks like. And they look generally very similar, which is to be expected. GFP's structure is pretty stable. But if we start to look at the difference between these two, we can start to see really important functional effects pop out. And so here, what I'm showing are in red, the enhancer specific contacts. So the contacts that are only present in the GFP bound to the minimizer nanobody or the enhancer nanobody, and then the minimizer specific ones bound to the minimizer. And so it would be really useful if we had some sort of model to actually predict the structural consequences of these types of binding interactions. So how might we go about doing that? If we think about two different amino acid residues, so here I'm showing two of the chemical structures, each for alanine, what we want is some model that takes as input those two residues and gives us back the probability of them being in contact with one another. And if we run this over all of the potential pairs of amino acids in a protein structure, we can generate this probabilistic contact map. And there are a bunch of different ways to do this. And in fact, AlphaFold has been very successful at generating these predicted contact maps for single chain structures. But one of the most promising, I think, is reframing this problem as a graph neural network problem. And so we can treat these proteins as a molecular graph because, as I said before, they're just a bunch of these amino acids chained together linearly. And so this, as I said before, is the structure of alanine. And what you might notice is that it looks a bit like a graph structure. You have your nodes and you have your edges that connect atoms with covalent bonds. And so if we apply graph neural network models, we might be able to actually model this process incorporating all of the atoms, not just the amino acids or the nucleotides like we did before for the FO4 and CBF1 modeling. And an important side note here is that using these atom level representations and not relying on the representations just of the amino acids as kind of elements of your alphabet is that it allows us a straightforward method for encoding post-translational modifications, or these chemical modifications that aren't amino acids but are added to the protein after it's been translated. And so in general, graph neural networks operate over these graph structures through message passing. And I tried out two different ones in very early stage development, these graph attention networks and position aware graph neural networks to perform this exact task, where we're going to input atom level features and the covalent adjacency matrix, perform graph convolutions for the two different residues, and then output the probability of contact, generating that contact map. And so we don't just generate a single contact map, but actually we generate different types of potential interactions. And so those of you familiar with chemistry might understand why we might want to predict hydrogen bonds or van der Waals interactions. We actually predict a set of 8 different types of interactions. And so this work is still very early stage, but I want to present some really preliminary results, just overfitting one of these graph neural network models to a single structure to see if it has the capacity to do that overfitting. And so if we look at one small protein structure here, we can use the different types of interactions. So here, hydrophobic, hydrogen bond, van der Waals, and water bridges as our target. And then if we try a bunch of different models, we can see varying degrees of success overfitting to that single structure, with one of the models actually perfectly fitting the data, indicating that this might be a feasible approach for modeling protein structure, and importantly, modeling the interaction of different protein structures, so complexes of different proteins. And so what I've shown you here is that these graph neural network models are really powerful and directly applicable and directly translatable to chemical prediction tasks. That by using all atom models of protein structure, we're able to predict the effect of post-translational modifications, or things that are added to the protein after it's been translated. And that by using a deep learning model to do this type of prediction, we'll be able to do it in really high throughput, allowing us to generate those protein libraries that I mentioned at the beginning of the talk that would allow us to do really high throughput screening for functional proteins. And so if we combine these types of methods with existing methods based on physics simulations and experimental processes, we can kind of find the needle in the haystack a lot quicker than previously possible. And with that, I want to acknowledge a bunch of people that were really integral in this work. So Dan Lai and Arjun were the 2 members of the Fordyce lab that really helped me develop the BET-seq assay. I got a lot of help from Anshul Kundaji's lab, in particular these individuals listed here, on developing some of the early neural network models for this. The Leskovic lab has been super helpful in shaping my thinking about how to apply graph neural networks to the protein structure prediction problem. And then these 2 collaborators down here at the bottom, Yaron and Thomas, were very helpful on some of the other graph neural network and some work that I didn't talk about. And with that, I'll open it up for any questions.
Great, thanks, Tyler. So I see one question, and people both on YouTube and people in the Zoom, can you please start asking your questions either in the Q&A or in the chat, and I'll come get you. The YouTube stream does run about 2 minutes behind, so we'll just take that into account. I do see one question here though, which is, what does the data for the neural network model look like?
So I'll assume, but correct me if it's wrong, that you're asking about what the inputs and outputs to that network look like. And so the input to the network is just the one hot encoded DNA sequence. And so it's just a 4 by L matrix where the individual rows represent A, T, G, and C. And then the length of that matrix represents the length of the DNA sequence. So that's the input. And then the output is the predicted delta delta G value. So that energetic value that we're actually measuring, we have a noisy estimate of that from our experiment. That's what we're using as the target for the training data.
Cool. This is not a question, but Daniel Cooper on YouTube says his mind is blown and he's really excited about the talk. Someone dabbed, so that was cool. I don't see, oh, I see another question. Have you been using any of the state-of-the-art NLP libraries, Hugging Face maybe, to parse and analyze the DNA sequences?
So we haven't been using that. And what a lot of the other groups at Stanford have found really successful are actually the 2D CNN models. So treating that 4 by L matrix, not as a sequence and applying NLP stuff, but applying a lot more of the computer vision techniques as a 4 by L image, basically. So they're using that to train and test networks over full genome sequences. So the sequences that I was looking at here were only 10 base pairs. They routinely train on thousands or hundreds of thousands long sequences.
Really cool talk. I was wondering, so it sounds like you think that the sort of 2D CNN approach is going to work better for these one hot encodings of DNA sequences. Do you have any thoughts about why something like an embedding based model isn't what works there, even though that's been so successful in NLP? And yeah, also, why do you think graph networks are so successful for the other tasks that you've used them for?
Yeah, so I think that the convolutional networks versus recurrent networks or transformers, I think that a lot of that comes back to not necessarily that they wouldn't be successful, but that the 2D CNNs they generate predictions in a way that's much more interpretable for biologists and for bioinformaticians. And so we're used to thinking about these things in terms of motifs. And if you think about each kernel in a CNN as basically a motif finder, it's going to apply a weight to each nucleotide at each position. Then people have already done a lot of the heavy lifting of kind of generating interpretation methods for 2D CNNs. So I think a lot of it stems from that. You might see greater success using some of the NLP techniques, but the other factor is that the data tend to be really noisy. And the trick is that depending on who collects the data, which group, how they do it, you run into a lot of these batch effects and things that aren't necessarily normalized over different experimental techniques or different experimental protocols.
Cool. And then Han asked, what type of linear models or techniques were used in the sequencing, and how has modeling the sequences using linear models helped in designing your neural network?
Yeah, so the linear models basically use the same inputs and outputs, and so it's just essentially a 40-dimensional vector where we're flattening that matrix. So it's the same one hot encoded matrix and the same delta delta G values, same inputs and outputs as the neural network models. And I'll have to admit that it didn't really shape our thinking about exactly how to design the neural network model. We kind of worked from the neural network model backwards. And so we built a neural network model that fit our data and was validated via these orthogonal methods. And then we used the linear models to really interpret what the neural network was learning and how that fit with our assumptions based on the data sets that had already been collected.
Nice. And then I think the last question is, have you tried experimenting with graph neural networks in addition to convolutional neural networks? And if so, is any of this stuff publicly available?
Yeah, so the graph neural network stuff that I presented at the very end is still really early stages. We're not seeing the same level of prediction accuracy yet as something like AlphaGo or these recurrent geometric networks. So if you're looking for the literature, I would point you to either the AlphaGo, or not the AlphaGo, the AlphaFold paper, or a lot of the work from Mohammed Al-Qarashi's group at Harvard does a really nice job with some of the recurrent networks. They seem to be more accurate in general so far, but I think there's promise for the graph neural network techniques.
Nice. All right, I think that's it. Thank you so much, Tyler. This was really cool.
Of course. Thank you, guys.
All right, up next, we have Kostov. So Kostov is a research PhD intern at Facebook AI Research, and his interests lie at the intersection of generative language modeling and